Part 2. Information within living systems. The genetic code. When examining the code, do we see patterns or design? Matter and energy or design and intent? The DNA, or deoxyribonucleic acid molecule in which the genetic code is contained, was discovered by Professors Watson and Crick in 1953. Some years later, they were both awarded the Nobel Prize for Medicine for their discoveries relating to information transfer in living material. In the next section we shall be identifying those features of the DNA molecule which qualify it as an information system. The judges of the Nobel Prize obviously thought that this was the case. Biochemistry is not for the faint-hearted. It's the most complicated thing on the planet. For this reason we shall be looking only at the information aspect of DNA. However, a little background must be attempted. DNA is an acid which has two backbones made of sugar and phosphate connected laterally by nucleotides. The whole forms a right-handed spiral. One of four types of acid bases attached to the backbone. The combination of these four bases encode the information. The four bases are called adenine, thymine, guanine and cytosine. Adenine only joins to thymine and guanine only joins to cytosine. A to T and G to C. An aid to memory is always toward God the Creator. Three of these characters make a three-letter word or codon. The genetic code reads these words along the backbone. These words specify amino acid chains, which then form protein chains, which then form all the parts of the living tissue that make up the organism. In our case, hair, blood, fingernails, bone, brain tissue, and so forth. The first observation then is that just like the sheet music and computer code, the genetic code does not resemble the end product. Okay, here are the words of the genetic code. Have a look at the words circled in black on the top left of the table. The first character C, second character T and third character T is the three-letter word or codon for leucine. I don't know anything about leucine, I'm just using it as an example. Notice there are 6 possible words for leucine, all having either C or T in them. This is our redundancy. Remember the mobile phone example? Even if a mistake is made in one of the characters, you will still get leucine. Given the 4-letter alphabet 3-letter word, it would be possible to code for 64 amino acids, but the system allows for 3 times redundancy by only coding for 20. There are many such error correction features in the genetic code. Let's use another example. The Morse code has a short pulse and a long pulse, separated by a space. These can be arranged to provide the 26 letters of the alphabet. From this we can produce the 400,000 or so words of the English vocabulary. In a similar way, the 4 nucleotides code for 20 amino acids and the 1 million proteins required to construct and maintain the human body. To summarize the vocabulary of the genetic code, nucleotide is equivalent to a character, codon to a word, gene to a sentence, chromosome to a chapter. Every cell in the human body has complete instructions for all the body parts of the individual. Okay, this is the last technical bit. The DNA we have been looking at is the library. For cell replication to occur, the information must be copied by a machine called a polymerase. This is a very complicated process, but sufficient to say a photographic negative is produced by unwinding the helix, making a copy, rewinding the helix, and returning it to the library. When a reverse copy of this negative is made, it will be the same as the original in the library. Amazingly, a man called Roger Kornberg has managed to photograph this in action. It is called transcription. He won the 2006 Nobel Prize for Chemistry for his achievement. The storage density of the DNA information is the highest in the universe. If you put a single coil of the helix on the head of a pin, it would be long enough to go 33 times around the Earth. If each codon was equivalent to a word, you could make 100 stacks of the average novel from the Earth to the Moon. The intent of all this information, of course, is to produce the amino acids which produce the protein chains, which are themselves subcomponents of larger systems. Very complex chemical bonds are required to cause the protein chains to be formed into the right shapes and to be in the right place at the right time. In 1949, the evolutionary biologist J.B.S. Haldane suggested that evolution would be falsified if complete structures were found in nature, such as the wheel, because a wheel is useless until it is fully formed. We now know that these complete structures do exist and are essential for the complex machines involved in cell reproduction, such as the RNA polymerase. Despite this evidence, the credulity of the atheist evolutionary establishment remains undiminished. We have already looked at the RNA polymerase. Consider the elegant design of this winding motor in a virus. The DNA is stored inside the virus. When required, the helix is wound out at tremendous speed. This is possible because of the design. The pink outer rotor pushes and the inner red rotor pulls. When the helix is returned to its stowage, the process is reversed. The two rotors must work in precise harmony, otherwise the helix would be snapped. A flagellum is a tail-like projection which is used to propel certain bacteria through a fluid, rather like a propeller. Rotating at 15,000 revolutions per minute, it can stop and reverse direction in a half rotation, thanks to a protein clutch which takes the motor off load, allowing the motor to reverse before re-engaging the clutch. This photograph of the motor is an electron microscope composite picture. Electron microscopes are necessary to view such small items, which are only nanometers across, because the wavelength of light is simply too large to provide definition. The drive motor has all the features of an electric motor. A universal joint connects the flagellum to the motor shaft, a bush passes through the inner and outer cell wall, the stator and rotor work through negative to positive electron flow. How can this elegant design and wonderful complexity possibly be explained in terms of naturally occurring processes? Surely it is more rational to consider these things in the light of a supreme designer and a creator. We've spent some time looking at the code for producing proteins, but this is only 3% of the code. The remaining 97% does not code for living tissue at all and is the operating system. This is rather like the word processor on your computer, the application program. It is much smaller than your Windows operating system, but you don't see the operating system. The American scientist Dr. Barbara McClintock made the discovery that living things are designed to adapt to their environment. She proved that damaged chromosomes repaired themselves by a process of transposition or genetic recombination, and that this was designed to conserve the code. For many years, her discoveries were not welcomed by the academic and scientific establishment. Why? Is it because they were chauvinist and didn't like female scientists? No, of course not. It was because the existence of an operating system directing and controlling the actions of genes disproves a central doctrine of atheist evolution, that of endless random mutations eventually giving rise to completely new kinds of life. This idea has never been seen to occur and can't be replicated in a lab. Little about this operating system is known at the moment, and many years later, despite Barbara McClintock's work, it is still called junk DNA by many evolutionists, said to be a leftover from our supposed evolutionary past. But as true knowledge is increasing, this assumption is being discredited. Barbara McClintock eventually received recognition for her outstanding work, but the knowledge that the action of genes is controlled is seldom discussed in the popular media.